Blame it on the goose, got you feeling loose, blame it on the drum.
So fermentation is called an electron dump. It's called this because its main purpose is to regenerate NAD+. And it's mainly used in the absence of oxygen because NADH is not oxidized in the electron transport chain when there's no oxygen left because oxygen is the final acceptor. So a problem comes up on how you can get NAD if the whole system is stopped up. So fermentation takes NADH and it oxidizes it, which means that it loses electrons to form NAD, so glycolysis can continue. Two main inputs of fermentation are 2 pyruvic acids and NADH, which was produced from glycolysis. And their outputs are lactate or ethanol. Ethanol is what's used in yeast, which we are using for the bread. And it goes from a pyruvic acid to 2-acetaldehyde, and then it gets converted to ethanol and carbon dioxide. What happens is the pyruvic acid goes in, NADH gets added to it, and so then it becomes this 3-carboned molecule with a hydrogen, and that'll either become lactate or it can become the acetaldehyde, and a carbon will come off to form carbon dioxide, and this will form the ethanol.
So glycolysis is the split of glucose. It's an overall exergonic reaction, and it's highly conservative, and it happens with or without oxygen, and it occurs in the cytosol. So you input glucose and you get 2 pyruvic acids, you input 2 ATP and you get 2 ATP, and you input NAD and you get 2 NADH. So the first part of glycolysis is the split of the glucose. The glucose is added to 2 ATP, and you get a glucose and a phosphate and 2 ADP because the ATP gave its phosphate to the glucose. Because they're so unstable, it splits into 2. So now you have 2 pyruvic acids. And then finally, the 2 pyruvic acids oxidize. So they lose a hydrogen, and that's 2 NADH. And they lose a phosphate, which equals 2 ATP.
Okay, ready? Oh, the smoke that just rose. Oh, beautifulness.